Welcome back friends, welcome again another video tutorial of A to Z biology. Today's discussion is lactose operon or simply called Lactoperon. First we will see that what is lactose operon. What is operon system? Operon system is a system in which different kinds of genes are arranged in close proximity under a single promoter and a single terminator. Suppose this is a gene, this is a gene, this is a gene, and this is the promoter region of that gene, and this is the operator region of the gene. This is gene 1, gene 2, gene 3, this is promoter, this is terminator. This arrangement is called polycystronic gene arrangement. It is only present in prokaryotes. In that scenario, in case of eukaryotes, under one promoter and one terminator, only one gene is present. But in case of prokaryotes, multiple genes are present. In case of E. coli is a gram-negative bacillus. In case of E. coli's DNA is circular and double-stranded. We all know that thing. If we isolate that DNA, here the structure of the DNA of a bacteria E. coli. This is the particular region of the DNA. Suppose this is the operon. Here the genes are arranged very close proximity, and these genes when expressed, they can produce some products which actually metabolize lactose. This is the fundamental concept of lactose operon. What is operon? And we will see that the picture of lactose operon. This is a DNA. This is the terminator portion. This is the promoter portion. P stand for promoter, T stand for terminator, and this is the operator. This is gene lac Z, lac Y, and lac A, and this is lac I. This is P lac I. You will be see that under a single promoter and terminator, multiple genes are present. This is Z gene, Y gene, A gene, and P stand for promoter, O stand for operator, and lac I stand for inducer, and this is the promoter of lac I, and this is DNA sequence. Previously I say that this portion is collected from the E. coli genome. What is the significance of this operon? If E. coli present in a medium, in that medium no carbon source is present for energy production, how E. coli can survive? E. coli cannot survive in that medium. But consider a situation, E. coli is present in a medium, in that medium glucose is present and lactose is present. E. coli always utilize glucose. It is the most preferable source of energy. But suppose another condition, E. coli is present in a medium in which no glucose is present, only lactose is present. In that situation, how E. coli can produce energy? How E. coli can survive in that environment? The answer is that the gene expression. E. coli activates its specific kinds of genes to produce a specific kinds of proteins, and by that proteins they can utilize lactose, they can break down lactose and form glucose and galactose, and by metabolizing them they can produce energy and they can survive in that environment. This is the very basic concept of Lactoperon. So Lactoperon is present in the prokaryotes E. coli. It is discovered by two scientists, Jacob and Monod, Jacob and Monod. These two scientists in 1961 discovered this Operon system, Jacob and Monod. So why this is promoter? This P region, this P region is the region at which RNA polymerase can bind. This P region is the region at which RNA polymerase can bind. RNA polymerase is an enzyme which helps in gene transcription. RNA polymerase bind with that promoter, transcribe the structural genes, and ultimately gene transcription leads to messenger RNA formation. Messenger RNA formation. And from this messenger RNA, when this messenger RNA is translated, these gene products will be formed. The Z gene codes for beta galactosidase enzyme. Beta galactosidase enzyme. Y gene codes for lac permease enzyme. Lac permease enzyme. And A gene codes for beta galactoside transacetylase enzyme. Transacetylase enzyme. Will we see that this function of these three enzymes in lactose metabolism? This beta galactosidase enzyme break down the lactose. Break the lactose. Lactose is a sugar. Lactose is a disaccharide formed by D-galactose and D-glucose. It is a disaccharide. Beta galactoside break down this lactose and form glucose and galactose. And E. coli can readily use that molecule and produces energy. And another function of the beta galactosidase is that this beta galactosidase converts this lactose into allolactose. Allolactose. Here, one important thing about the lactose is that lactose is an inducer. This system is an inducible system. Inducible system. Why lactose is an inducer? If the lactose is present in the growth medium, E. coli growth medium, if the lactose is present in the E. coli growth medium, this all scenario will be activated. This gene transcription will be occurred, this enzyme production will be occurred, and all these things will be happened. So lactose is an inducer. Lactose is an inducer, but the most powerful inducer is an allolactose. Allolactose. Allolactose is an isomer of lactose, which is produced by beta galactosidase. See that normally this gene transcription will not occur, because normally E. coli do not need any kind of these three enzymes. Suppose consider E. coli is present in a medium in which glucose is very abundant. So they do not need any kind of gene transcription, any kind of this activation, this huge phenomena. So they can simply use glucose, and after the depletion of glucose, when glucose is no longer present in that medium, they will activate this enzyme. So in all the time, this LAC-I gene produces a messenger RNA, a mRNA, messenger RNA. When this messenger RNA gets translated, this messenger RNA forms a protein product. That protein product is called repressor protein. Normally this repressor protein forms a tetramer, and this repressor protein binds with the operator region of this operon. And prevents the binding of RNA polymerase and prevents this transcription of these three structural genes. So normally this LAC-I gene is always expressed. This is constitutive genes. In eukaryotes, in prokaryotes, some genes are always expressed. These genes are always called constitutive genes. And some genes are expressed depending on the cells need, depending on the organism's need, those genes are called non-constitutive genes or regulatory genes. So in that case, lac-I gene is constitutive gene. Lac-I gene always codes, and lac-I gene always codes this repressor protein, and this repressor protein forms a tetramer, and this repressor protein bind with the operator and prevents the binding of RNA polymerase. So these genes cannot be transcribed in normal case, and this is the promoter of lac-I gene. So as soon as lactose is present in the medium, beta galactoside, lactose is present in the medium, lactose will be converted into allolactose. This allolactose can bind with this repressor protein and converts this repressor protein, and repressor protein gets altered. Repressor protein size get altered, so it no longer be able to bind with the operator region. So these genes can, RNA polymerase can bind, and these genes will be transcribed, and ultimately 3 enzymes will be produced. But one important thing about that, this is suppose E. coli cell. First function, we know that beta galactosidase actually breaks the lactose and converts some of the lactose to allolactose. Second enzyme function is that lac permease. Lac permease actually allow the lactose to enter into the cell. This is the most important point. Please focus it. Lac permease allows the lactose to enter into the cell. If lactose is not entered into the cell's cytosol, how this all operon system gets activated? This is very confusing. If lactose is present in the medium, that's it. But if lactose does not present in the cytosol, how it can initiate this whole process? The answer is that these all enzymes are always present in the E. coli cell in very low amount, So as soon as lactose comes in the medium, glucose is depleted, glucose is no longer present, and lactose is just comes, then lactose get entered into the cell due to presence of this lac permease protein, and lactose as soon as enters the cell, this gene transcription will be more and more this product will be formed. This is the actual concept and modern concept. In the presence of glucose, in the presence of lactose, all the time E. coli cell possess 3 enzymes in very low concentration, because this repressor, I previously said that you will be remembered that this repressor is bind with this operator and prevents this binding of this RNA polymerase. This is RNA polymerase. But this binding is not permanent binding. Repressor is bind with the operator and prevent the binding of RNA polymerase, but this binding is not permanent binding. They can bind and dissociate. And at that time RNA polymerase will be bind and transcribe these sets of genes. So very low amount of 3 enzymes is always present in the E. coli cell. But we know the functions of 2 enzymes. Another enzyme is actually beta galactoside transacetylase. They can transfer acetyl CoA to beta galactoside. They can transfer acetyl group from acetyl CoA to beta galactoside, but the actual role of this third enzyme in the Lactose system is not still clear. So this is the most important phenomena of Lactose, and this repressor binds with operator. This is the negative regulation of Lactose. Another type of regulation is positive regulation, and you will be see that positive regulation, how this operon system is regulated, how this operon system, negative regulation, positive regulation work coordinately in the presence of lactose, in the presence of glucose. You will be see that in the next video lecture. The last portion of this section is that lactose is an inducer. Lactose is a natural inducer. Lactose is present, this operon system is on. Lactose is absent, this operon system is off. Lactose is present, glucose is present, this operon system is off. Even in the presence of this IPTG, IPTG is a synthetic inducer. This is a synthetic molecule, isopropyl beta-1 thiogalactopyranoside. This molecule mimics the structure of allolactose and activates this whole gene transcription, whole gene activation, whole gene expression actually. So this is an synthetic inducer. This is an synthetic inducer. This is the most important for question point of view. This is a synthetic inducer. This is natural inducer of this operon. IPTG, isopropyl beta-1 galactopyranoside is an synthetic, thiogalactopyranoside is a synthetic inducer, and this is natural inducer.